My name is Farnaz Kadem. I'm Stanford's Vice President for University Communications, and I'm absolutely delighted to welcome you to the press conference in honor of our newest Nobel laureate, Professor Carolyn Bertozzi. This morning, Professor Bertozzi, along with Morton Meldal and K. Barry Sharpless, was named as a recipient of the 2022 Nobel Prize in Chemistry for the development of click chemistry and bioorthogonal chemistry. Today, we have with us Mark Tessier-Levine, President of Stanford, Deborah Satz, Dean of the School of Humanities and Sciences, Lloyd Minor, Dean of the School of Medicine, and of course, our laureate, Professor Bertozzi. Before we get to your questions, we'll start with a few remarks from our speakers. And for members of the media in our audience, if you've not already done so, please submit your questions now using the box at the bottom of the webcast page. We'll get to as many of them as we can, and a recording of this discussion will also be available at the conclusion of the conference. So with that, it is my pleasure to introduce Mark Tessier-Levine. Mark.
Well, thank you, Farnaz, and good morning. I want to thank all of you for joining us this morning as we congratulate Carolyn Bertozzi on her remarkable achievements. All of us at Stanford are just so incredibly proud of Professor Bertozzi, and I'm so delighted that her accomplishments have been recognized by the Nobel Committee. In pioneering the field of bioorthogonal chemistry, Carolyn invented a new way of studying biomolecular processes, one that has really helped scientists around the world gain deeper understanding of chemical reactions in living systems. She tailored the work that her fellow laureates, K. Barry Sharpless, a professor at Scripps Research, and Morton Meldal, a professor at the University of Copenhagen, were doing in click chemistry. And she tailored it to create a novel way to apply the reaction to biomolecules in living systems without affecting their chemistry. The real-world impact of her work has already been profound and far-reaching, revealing how cells build proteins and other molecules, unleashing new diagnostic and therapeutic approaches to treat disease, and producing new materials for energy storage, among other applications. In her own laboratory, Carolyn has applied this method to answer fundamental questions about the role of sugars in biology and their impact on underlying diseases, including cancer, inflammation, tuberculosis, and COVID-19. She's used these discoveries to help develop new diagnostic and therapeutic approaches, notably in the area of immuno-oncology. As we consider the challenges of our world, including chronic and emerging diseases, Carolyn has revolutionized our ability not only to understand the science of disease, but also to create interventions that have the potential to impact lives around the world for the better. Carolyn will describe her work in fuller detail in just a few moments. But before we move on, I do want to add that I'm inspired not only by Carolyn's remarkable contributions to science, but also her contributions here on campus at Stanford. She's a prolific mentor, advising more than 250 undergraduates, graduate students, and postdoctoral fellows over the course of her career. And as director of Seraphan ChemH, Carolyn's built strong connections between the Institute and fundamental researchers in chemistry and biology, as well as clinical researchers based in Stanford Medicine. So I'm so grateful to all that she's given to our university and to the world. This win follows in a great tradition. Carolyn is in good company. She is the 21st living Nobel laureate among Stanford's community of scholars. There have been 36 Stanford Nobel laureates in total over the years, including last year's winner of the Nobel Prize in Economics, Guido Imbens, as well as the 2020 Nobel laureates in economics, Paul Milgram and Robert Wilson. So thank you again for joining us this morning to congratulate Professor Bertozzi for her remarkable scholarship. I'm inspired by her work, and we are all deeply grateful for her contributions. And now I'd like to turn it back over to Farnaz. Farnaz?
Thanks, Mark. And I would like to turn it back over to Professor and Dean Deborah Satz. She's the Dean of the School of Humanities and Sciences. Deborah.
Hello. It's wonderful to be here to celebrate the extraordinary honor that Carolyn, along with two other amazing scientists, have received today. Carolyn has been at Stanford and in the Department of Chemistry since 2015. And she's been instrumental in shaping Seraphan ChemH, institute that brings together, as Mark described, chemists, biologists, engineers, and clinicians to better understand life at the molecular level with the aim of improving human health. And obviously, this is a perfect match of her work and her interests. You couldn't put together, and it's no accident, because she really was instrumental, along with Chetan Khosla, in shaping ChemH. She has a BA and a PhD in chemistry, but her work really straddles chemistry and biology. She actually started as a biologist, but switched to chemistry. Also, I just have to note that she almost majored in music, almost had a career in music, and sang backup vocals in a heavy metal band called Board of Education during college. She's won almost every award for groundbreaking research in the sciences you can think of: the Wolf Prize, the Lemelson MIT Prize, and a MacArthur Fellowship. And her work does what great science does. It illuminates the world. It throws light on how our cells work, which in ways that have profound implications for the treatment of cancer and disease. She's recognized for creating a new field of bioorthogonal chemistry, which again, as was noted, develops techniques to tag complex sugars in our cells in order to understand what the sugars are doing in ways that don't disrupt the function of cells while doing it. And that's hard science to do. And this has incredible applications for human health. Not only did she discover an entirely new field of chemistry, but she's also a dedicated and inspiring teacher. She teaches introductory undergraduate chemistry courses, and chemistry is often not students' favorite subjects, but she is a favorite teacher at Stanford. She's mentored hundreds of grad students, postdocs, and colleagues. And she's also been a longtime champion for increasing diversity in the scientific profession, and co-founded with her colleague Chetan Khosla the postbaccalaureate program in target discovery to prepare college students from diverse and historically underrepresented backgrounds to apply for doctoral programs in the sciences. She's received the prestigious Lifetime Mentor Award from the American Association for the Advancement of Science just recently in 2022. Her extraordinary work is a testimony to the critical role that research universities like Stanford play in blue sky research that's high stakes, that's unfettered by anything except energy, creativity, and imagination, and that creates new knowledge that benefits all of us. So congratulations, Carolyn. We're just delighted, so proud. Basking in your glory.
Well, first of all, Deborah, that's a tough introduction to follow. and Mark, thank you so much for supporting ChemH and everything that we're doing at Stanford. There aren't that many occasions where your bosses get together and say nice things about you. So this is definitely very special for me. I really appreciate it. And Farnaz, thank you so much for organizing this and pulling us all together.
Well, we couldn't be more thrilled for you, Carolyn, and all of your accomplishments. I would love to have one more person give their thoughts and share their accolades with you, and that is Lloyd Minor, the Dean of the School of Medicine. So Lloyd, I'll turn it over to you.
Well, thank you very much, Farnaz. And Carolyn, congratulations on behalf of all of us. I'll be brief. Mark and Deborah have done such a wonderful job in describing your work, Carolyn. I've been inspired by your work since we first met when we were in the process of bringing you to Stanford in 2015. And learning about your approach, and which is so well established. You and your colleagues have so well established in the Seraphin ChemH Institute, learning about your approach of bringing together fundamental chemistry with principles of engineering, and of course, a deep understanding and appreciation for biology and the impact of that approach in, for example, our understanding of the basic building blocks of life, our understanding of the glycoproteins, the sugars that coat all cells, and how you've brought that knowledge to improve diagnostics in diseases such as tuberculosis, and also to develop entirely novel approaches to therapeutics, including, very importantly, cancer therapeutics. I also want to emphasize, Carolyn, what an amazing colleague you are. Your vision, your insight inspires all of us. As Deborah mentioned, you've mentored scores of graduate students, postdoctoral fellows, that each in their own right are now doing transformative work, and that each have benefited so much from your knowledge and from your dedication. So Carolyn, on behalf of all of your Stanford colleagues, your scientific colleagues around the world, congratulations on this very well-deserved award. And we just look forward to learning from you, working with you, having the privilege of your science and the privilege of you as a colleague for many years to come. Congratulations.
Thank you so much.
Thank you, Lloyd. Much appreciated to all of our speakers. And now I would actually like to ask a few questions of our newest laureates. So Professor Bertozzi, it's been quite a morning for you. Let's start with the most obvious question. What's it been like? What happened this morning when you got the call? What have the last few hours been like for you?
Well, it's crazy. I mean, it's a whirlwind. And, you know, it started with a jarring phone call at, I think it was 1:43 a.m. I took a screenshot of my phone. And I was asleep like a person should be at that time. And I woke up and, you know, all of a sudden someone introduces himself as a member of the Nobel Committee calling with some good news. And you sort of have to pinch yourself a few times to make sure it's not just a hallucination in the middle of the night. But basically what they told me was information that I don't remember. It was a blur. But then the last thing I remembered was they said, you have just under 1 hour before the announcement goes live on the live stream. So enjoy the last hour of your previous life, because then it will start your new life. And that's what this is. So it's just been, I'm overjoyed. You know, I've been on the phone and on Zoom for the last 6 hours or so, connecting with people from the media. And I've had lots of very kind congratulatory emails and text messages. And I think all 250 of my former students and postdocs must have emailed me in the last few hours.
So everybody's wanted to wish you congratulations. That's wonderful to hear. We have a lot of questions from those joining us on the live stream to hear from you directly about your research. So let me just read a few of them and then turn it over to you to maybe talk a little more about this. So there's one that says bioorthogonal chemistry is often praised for being simple and elegant. So what is the simple explanation of how it works? Bioorthogonal chemistry depends on a 100-year-old chemistry reaction that your group rediscovered and repurposed. Can you talk about that? And then one that asks, how did you come up with the word bioorthogonal?
Okay. Well, let me do them a little bit in somewhat reverse order. First of all, these are fantastic questions. Whoever posed those questions has been paying attention. So bioorthogonal literally means, to the scientist, not interacting with or interfering with biology. So orthogonal is the not interacting with or interfering with part, and bioorthogonal means not interacting with biology. So what that means in practice is that we basically develop pairs of chemical groups, and those pairs of groups are perfectly suited for each other. And when they encounter each other, they want to react and form a bond. And they love each other so much that you can surround those chemical groups with thousands of other chemicals. And that's what you have in biological systems, in your cells, in your body. There's thousands of chemicals. But these two chemicals that are bioorthogonal will ignore all of that, and they'll find each other and form a bond with each other, do chemistry with each other. So that's actually incredibly useful if you want to use these chemical tools to study a biological process. And before the advent of bioorthogonal chemistry and then the related chemistry that Professor Sharpless and Meldal developed, which is they call click chemistry, before these chemistries were available, there was really no way to study certain biological processes. They were just invisible to the scientist. But these chemistries make those processes visible, and we have benefited from that specifically to study cell surface sugars. That's the area of biology that my lab focuses on.
Great, thank you. What inspired you to go into this field of research?
Well, you know, I would say my interest in chemistry, broadly speaking, started in college. And I was a pre-med for a while, like so many undergraduates declare themselves. And as a pre-med, you're required to take a collection of introductory science courses. And organic chemistry is one of them. And it's probably the most notorious of them because organic chemistry has a reputation for being difficult, impenetrable, kind of a weeder course for pre-meds that many would complain later on, keep them out of med school ultimately. And so I went into that class fully expecting, you know, to just grind it out and suffer like everyone else. But unexpectedly, it was an amazing course that just really resonated with me. And I just fell in love with the subject. And so by the time I graduated from college, I had embraced myself, my identity as an organic chemist. And I knew that that's what I wanted to do. And then as a graduate student, I practiced organic chemistry around sugar molecules. And that's where I got interested in the biology of sugars. And so when it came time for me to think about what would I do with my own independent research, I had this love of sugars and the biology of sugars, and I wanted to learn more about that. And at the same time, I loved organic chemistry. And it turns out I could bring those two things together. And so bioorthogonal chemistry was a tool that my lab created originally to study cell surface sugars. In fact, to image cell surface sugars using microscopes. But then it turned out to be so useful just as a platform for studying biology that lots of other labs picked up on it and started using those same chemistries to study other molecules like proteins, DNA, and RNA. And it turns out you can study these molecules in live cells and in laboratory animals. And the most exciting development is now there's a pharmaceutical company doing these chemistries inside the body of human cancer patients as a means to deliver drugs to cancers. So the field has really progressed a long way in the last 25 years, and it's very exciting for me to see this.
So your last point there led exactly to the question we just got, which says, how can your tools be used to fight cancer?
Well, so right now, there are two, I'm thinking at least two, maybe three companies that have made medicines with bioorthogonal chemistry to treat cancer. The first generation of those medicines are called antibody drug conjugates. And one of the companies pursuing that is one that I co-founded from my laboratory. But even more recently, there's another company called Shasky Pharmaceuticals. I'm on their scientific advisory board. What they do is they will take one of the bioorthogonal reaction partners and inject that into a tumor. And then they infuse the other bioorthogonal reaction partner into the circulation of the patient. And that second partner has a drug on it. And when the second partner finds the first partner and reacts, the drug gets released at very high doses, very concentrated in the tumor. And so that allows the oncologist to treat the tumor and kill the tumor without exposing the person's entire body to a toxic chemical. It's amazing. And they're in a phase 1 clinical trial right now.
That does sound absolutely fascinating. And it just really, I think, demonstrates the real world applications of the work you've done, which I know is something you're particularly been interested in. There's a question here that I think is really interesting. How did failures lead to some of your biggest successes?
There's a fine line between failure and success, as we all know, because many of us define failure as, an outcome of an experiment that is not the intended outcome. But of course, when the outcome is not the intended outcome, that doesn't mean it's a failure. That means you learned something unexpected. And the question is to understand, what have you actually learned? And we've certainly had many unexpected results along the way in developing our reactions. But I would say one of the most interesting recent, failures turned successes was we were using bioorthogonal chemistry to try to understand the kinds of sugars that are on surfaces of cells. And unexpectedly, we discovered a whole new type of molecule. And so sugars are anchored to cells either through their attachment to lipids or to proteins. And so as far as the world knew, all of the cell surface sugars took the form of glycolipids or glycoproteins, where the glyco means sugar. And with bioorthogonal chemistry, we discovered there's a whole third type of glycomolecules on cells called glycoRNA. And that was a total mind blow, because nowhere in the textbooks of cell biology would it teach you that it's possible to make a glycoRNA and to put it on the surface of a cell. But undeniably, these molecules exist and they are everywhere on our cells. And we found them by using the spotlight of bioorthogonal chemistry.
Terrific. Thank you. Let's switch over to something that all of our speakers have mentioned, which is obviously your passion for mentoring and the hundreds of students that you've mentored through your career. And there's a couple of questions that are related to that. One is just, why do you feel that is so important? Why have you clearly spent so much time and effort to mentor so many students throughout your career? And then I think a related question is, you've spoken about how your mentors and parents encouraged you, and can you share how they influenced your career? So I think two sides of the same question.
Yeah, absolutely. I think all of us who are academics, and especially in the sciences, know that progress in science is basically a process of incremental accumulation of new knowledge and generation of new inventions as we stand on the shoulders of those who came before us and established the precedents. And so it's very important to me that I have the opportunity to mentor scientists, younger scientists, because I know that whatever my lab can contribute to the world, it's a sliver compared to what the totality of all of my mentees will contribute to the world. So mentoring students gives you an opportunity to amplify the impact of your science, because the knowledge that they accumulate from working with you becomes the kernel of a whole new scientific enterprise in their own hands down the line. So that really is the cycle of science. It's being mentored and then mentoring, paying it forward and giving people a foundation that they can use to build new knowledge. Also, personally, I find it very rewarding to work with young scientists, because I think back to the feeling I had when I was a young student, not sure about what I would do with my life. And as I mentioned before, I took this one, fated class, organic chemistry, had an amazing professor who brought the subject to life for me and changed my life. And I remember the exhilaration I felt when I had that epiphany that this is what I was meant to do. And I really hope that I can provide that kind of joy and opportunity to other people as it was provided to me.
And so on that note, I wanted to ask you about the two professors with whom you share this prize. And I would love to hear, and I think others would as well, you built on their work. What is it? How does it feel like to share this prize with them? Have you spoken to them this morning?
I haven't, I emailed them both immediately before it went public. I know them both very well. Barry Sharpless is at Scripps in La Jolla. And, he and I have known each other for basically since the beginning of my career. But even before that, I was well aware of his work because he was already very famous as a senior organic chemist with a Nobel Prize. This is his second Nobel Prize. His first Nobel Prize was for developing what's called asymmetric catalysts, and it was legendary work. And I studied his work when I was a graduate student. So he was already, a person who, was very inspirational to me. And then when I was in graduate school, I read the papers published from Morton Meldal's group, and he was a young professor at the time. He was doing peptide synthesis and glycopeptide synthesis. So he was another, legend of mine, a luminary whose work I admired. And so here we are. I don't think any of us would have anticipated that 25 years later, we would share this kind of a stage, but to be included in the same breath with those two chemists is a huge honor for me. Absolutely.
And it seems like it makes it all the more special since you've known them for so long and worked with them. So
yeah, and it's even better than that because we have a relationship where, I've had graduate students from my lab become postdoctoral fellows in Barry Sharpless's lab. And likewise, I've had a student from his lab as a postdoc in my lab. So we've had people exchanges as well as scientific exchanges.
No, that's great. Thank you for that. Let me ask you, we wrote in the story that we published about the prize this morning that your father is a retired physics professor, and he was the first one you called this morning.
Yeah,
what was his reaction?
right.
What's been the reaction of your family?
Well, as you might imagine, my dad was just blown away and just bursting, with excitement and joy. He's 91. He retired from MIT when he was about 80. And at that time, he actually started his second career. He founded a company and sort of worked on that for a decade. But then a year ago, my mother passed away, and they lived in the Boston area all of their lives. That's where I grew up. And so we actually moved my father out to California. So now he lives in Los Angeles, just a few miles away from my older sister, who's a professor at UCLA in applied math. And when we moved him out, it was a whole new life, a whole new chapter for him. But at the age of 91, he wasn't really sure if there'd ever be another monumental event in his life until today. And so when I called him, first of all, he was awake. He's a night owl. He was watching TV at 2 a.m. And I said, Dad, well, first of all, I think when a child calls you at 2 a.m., usually it's bad news. But in this case, I said, Dad, you won't believe I just had a phone call and you won't believe what it was. And he paused for a moment and then he guessed it. So, yeah.
That's incredible. That's good. That's amazing. What a wonderful thing to be able to share with him at 2 o'clock in the morning.
I'm so happy that he is here to see this. Yeah.
I can imagine. Let me ask you one final thing, which is there anything you would want to say we have not talked about, either about your research or any other thoughts you'd want to share with everyone who is watching this from around the world?
Oh, gosh. Well, we've talked about a lot of things, but maybe it's a good time for me to articulate my gratitude to the many students and postdocs who took a chance and joined my lab. Many joined my lab at a time when I was brand new professor with no track record and no reputation, and proposing to do all kinds of crazy things like do chemistry in cells and in people and so on. But enough of them, were fierce enough and fearless enough and intrepid, and they did this beautiful work. So I guess, here I am as a vehicle to share the joy of bioorthogonal chemistry, but they're the ones who deserve the credit. So hopefully that's a message that gets out there.
Well, I'm sure that they are all as incredibly proud of you as everyone here at Stanford is. I also understand that there's another special event next week. It's your birthday. So this is a nice early birthday present, I hope. Gives you another reason to celebrate.
It sure does. Yeah. I normally wouldn't celebrate a birthday at my age, but
well, hopefully this one is one especially.
Maybe I'll make an exception this time. Mm-hmm.
Well, many congratulations from all of us, Professor Bertozzi, and we wish you all the best. We couldn't be more proud. And I want to thank you for all the time that you've spent this morning with my team and talking to people. I want to thank all of our speakers, Mark, Deborah, Lloyd, for joining us. And I want to thank all of you who've joined us from around the world. And I wish you a very good day. Thank you.
Thank you all.